So let us start our today's topic. We were discussing immunity and we are discussing adaptive or specific immunity. In the last lecture, I told you about the four distinguishing properties of adaptive or specific immunity. So first of all, it only works in the presence of the pathogen or disease. Number second, it displays a specific response against a specific pathogen or a specific disease. Number third, it displays memory. It keeps record of the invaded pathogen. So the pathogens which had invaded our body, their record is kept by this type of immunity. And number four, it seldom attacks its own body components. So when something goes wrong, this adaptive or specific immunity, it may attack its own body components, causing autoimmune disease. Now today we are going to discuss about the cells of this adaptive or specific immunity. Where from we get this adaptive or specific immunity? In our bone marrow, in our bones, there is a bone marrow known as red bone marrow. There is a type of bone marrow known as red bone marrow. Then we also have got white bone marrow. White bone marrow or yellow bone marrow that is present in the marrow cavity. So here we have got yellow bone marrow or white bone marrow. This is yellow or white bone marrow. This is mostly stored fat. Well as at the ends of the bone, we have got a red bone marrow. Red bone marrow. This red bone marrow, it has got some stem cells in it, from which all the blood cells are produced. So from this red bone marrow, in this red bone marrow, there are progenitors. There are progenitors of lymphocytes. There are progenitor cells of lymphocytes. That means there are those cells which will give rise to lymphocytes. So lymphocytes, they are developed in red bone marrow. Then some of these lymphocytes, they come out of the red bone marrow and they migrate to thymus gland. Thymus gland, where these lymphocytes they become mature. Where these lymphocytes acquire maturity. So those lymphocytes, after arising from the bone marrow, which migrate to thymus gland and mature there, those lymphocytes are known as T lymphocytes. They are known as T lymphocytes. And there are some lymphocytes which do not come out of the bone marrow. They remain in the bone marrow. They reside in the bone marrow. Or they come out of the bone marrow and go to lymphoid tissues of the body and mature there. Lymphoid tissues. And they mature there in the lymphoid tissues, lymph glands, right? Those lymphocytes, which either remain in bone marrow and mature there, or come out of the bone marrow and go to lymphoid tissues and mature there, those lymphocytes are known as B lymphocytes. So lymphocytes, they arise from the red bone marrow, like RBCs, like WBCs, like platelets. Lymphocytes also arise from red bone marrow. The cells which give rise to lymphocytes, they are known as progenitor cells of lymphocytes. So once these progenitor cells start forming lymphocytes, some of the lymphocytes they come out of the bone marrow and they migrate to thymus gland and mature there and mature there. Those lymphocytes which mature at thymus gland, they are known as T lymphocytes, or we call them as thymus dependent lymphocytes. Thymus dependent lymphocytes. We call them as T lymphocytes or thymus dependent lymphocytes. Some of the lymphocytes, they reside in the bone marrow, they mature there, or some of them come out of the bone marrow and mature in lymphoid tissues, other lymphoid tissues of the body. Those lymphocytes which mature in bone marrow or other lymphoid tissues of the body, they are known as B lymphocytes. We call them as B lymphocytes. Now, these T lymphocytes and B lymphocytes, on stimulation by antigen presenting cells, they change into T lymphoblasts and B lymphoblasts. Now, these T lymphocytes and B lymphocytes, on stimulation by antigen presenting cells, I need to rub this diagram. Now, T lymphocytes and B lymphocytes. On antigen presentation, on antigen presentation by APCs, antigen presenting cells. So when APCs, antigen presenting cells, present antigens to T lymphocytes and B lymphocytes, T lymphocytes, they change into T lymphoblasts, and B lymphocytes, they change into B lymphoblasts. A blast, when we use the word blast means dividing cell. So this T lymphocyte, it starts divisions. And this actively dividing T lymphocyte, it is now known as T lymphoblast. B lymphocytes, they also start division. And this actively dividing B lymphocyte, it is known as B lymphoblast. So these T lymphocytes and B lymphocytes, when they are stimulated by antigen presenting cells, they change into T lymphoblasts and B lymphoblasts. Then T lymphoblasts gives rise to three types of cells: killer T cells, helper T cells, and regulatory T cells. This is killer T cell. They change into killer T cells. They change into helper T cells. And they change into regulatory T cells. Regulatory, or we also call them as suppressor T cells. Suppressor T cells. So T lymphoblasts, they divide and they give rise to three types of T cells, known as killer T cells. We write it as TK cells, killer T cells, helper T cells, and regulatory T cells. So killer T cells, helper T cells, and regulatory or suppressor T cells. Similarly, B lymphoblasts, they divide into two types of cells: plasma cells and memory cells. Plasma cells and memory cells. B lymphoblasts, they divide into two types of cells known as plasma cells and memory cells. So what is the role of these cells? These cells provide us adaptive or spastic immunity. These cells provide us adaptive or spastic immunity. So how do they provide us adaptive or spastic immunity? Killer T cells, as the name indicates, they kill the pathogens. They kill the pathogen. Killer T cells are those lymphocytes which kill the pathogens. So how do they kill the pathogen? They kill the pathogens in three ways. by releasing perforins. Perforins. Perforins are the defensive proteins which punch holes in the cell wall of the pathogen and destroying the pathogen. So these perforins, they are the proteins released by killer T cells. And these proteins, they punch holes in the cell wall of the pathogen. They punch holes. They punch holes in the cell wall of pathogen, killing the pathogen. So perforins are released by killer T cells. When they release perforins, these perforins, they are proteins, and they punch holes in the cell wall of the pathogen and they destroy the pathogen. Or they release, or these killer cells, they release granzymin, and granulosin, and granulysin. These are again proteins, defensive proteins. These are again defensive proteins. And these defensive proteins, they induce programmed cell death. They induce programmed cell death in pathogen. Or we say they induce apoptosis. Apoptosis in the pathogen, right? So killer T cells, these are the T lymphocytes which are involved in killing the pathogen. So how they kill the pathogen? They kill the pathogen in three ways. They release perforins. These perforins, they are defensive proteins which cause holes in the cell wall of the pathogen. So thereby bursting open the cell and destroying the pathogen. Or they release granzymins and granulysins. These are defensive proteins which enter through the cell wall into the pathogen, and then they induce programmed cell death in the pathogen. They start degenerating the DNA of pathogen. They start degenerating the cellular machinery of the pathogen. So over a period of time, the pathogen internally dies. Over a period of time, the pathogen automatically dies. So this is known as programmed cell death. They mostly affect the genetic material of the pathogen. And that change in the genetic material of the pathogen, that induces death in the pathogen cell. And we call this as apoptosis. Apoptosis, right? So this is the function of killer T cells. So as the name indicates, they are a type of T lymphocytes which kill the pathogens by perforins, by granzymin, by granulysins, right? So they are involved in the destruction of the pathogen. They kill the pathogen. That is why we call them as killer T cells. Then we have got helper T cells. Helper T cells. Helper T cells are a type of T lymphocytes which are involved in the activation of the other immune cells. They release some proteinous chemicals. Known as cytokines. So helper T cells, they release some defensive proteinous chemicals known as cytokines. And these cytokines, they stimulate both T lymphocytes and B lymphocytes for destroying the pathogen. You can understand the role of helper T cells by studying AIDS. You know, in AIDS, a patient suffers from total lack of immunity. Why in AIDS, a patient suffers from total absence of immunity? Because this HIV, it destroys the helper T cells of a person. HIV, human immunodeficiency virus. It resides in the helper T cells. It multiplicates in the helper T cells. So in the process of multiplication, it destroys all helper T cells of the patient. So when all helper T cells of the AIDS patient are destroyed, these helper T cells are not able to stimulate T lymphocytes and B lymphocytes. So they also serve as antigen presenting cells for T lymphocytes and B lymphocytes. They stimulate T lymphocytes and B lymphocytes for generation of other defensive cells. So when helper T cells are destroyed, then T lymphocytes and B lymphocytes, they are not stimulated. They are not stimulated, and immune system is shut down. AIDS, as I have told you earlier, it is not a disease itself. AIDS is simply a case of immunocompromisation. In AIDS, a person simply gets sick because of non-functioning of the immune system. So when immunity is not functioning, any opportunistic infection attacks the suffering patient, right? So in AIDS, the main problem is destruction of helper T cells by HIV. So when all the helper T cells are destroyed, then immune system is not stimulated. So for stimulation of immune system, we require helper T cells. Because APCs, antigen presenting cells, they mainly present antigens to helper T cells. Then helper T cells present these antigens to T lymphocytes and B lymphocytes, right? So APCs, antigen presenting cells, they first present the antigens to helper T cells. Then helper T cells, they present the antigen by cell-cell contact or by releasing cytokines to T lymphocytes and B lymphocytes so that they are stimulated, right? So these helper T cells, either they directly serve as antigen presenting cells and they present antigens to T lymphocytes and B lymphocytes, or they release these proteinous chemicals known as cytokines, which stimulate T lymphocytes and B lymphocytes for generation of an immune response. If helper T cells are destroyed, the immune system is destroyed. If helper T cells are destroyed by virus, then the whole immunity is shut down because it is the helper T cell which stimulates T lymphocytes and B lymphocytes to change into B lymphoblast and T lymphoblast, right? To form other types of defensive cells. So the key of our immune system is the helper T cell, right? So antigen presenting cells, they produce their antigens to helper T cells. Then helper T cells may serve as antigen presenting cells for T lymphocytes and B lymphocytes. Then B lymphocyte will change into B lymphoblast. T lymphocyte will change into T lymphoblast. Then it will generate killer T cells, more helper T cells than regulatory T cells. However, if the T cell is destroyed, then T lymphocytes and B lymphocytes, they will not change into T lymphoblast and B lymphoblast. Then other immune cells are not produced, right? So these cytokines, cyto means cell, kines means movement. So they cause the they cause the interaction of different cells. So these are proteinous chemicals which stimulate the other cells for action, right? These cytokines, they involve different types of chemicals. They involve interleukins. Interleukins, it is a type of cytokine, right? There are different types of interleukins. Interleukin 2, IL-2, IL-3, IL-4, IL-5, IL-6, right? We write it as IL-2, IL-3, IL-4, IL-4, IL-5, interleukin 5, interleukin 6, right? So they release different types of interleukins which stimulate the action of the T lymphocytes and B lymphocytes, right? They also release interferons. Helper T cells also release interferons. Interferons are the proteins which are used for inactivation of the virus. They do not allow the virus to enter uninfected cells. So they protect the entry of virus into the cells. Or sometimes they bind to the virus and do not allow the virus to enter into the cells. Or sometimes they coat the uninfected cells and do not allow the virus to enter into that cell. So they inactivate the viruses. Interferons inactivate the viruses. These helper T cells, they also stimulate. they also release TNF, tumor necrosis factor. So wherever a tumor may be developed in our body, these helper T cells, they release chemicals known as TNF, tumor necrosis factor, that destroys the tumors, right? So they do not allow the cancers to develop. They destroy the cancer, they destroy the tumor, right? And these helper T cells, they also release monocyte colony stimulating factors. Stimulating factors. So they also stimulate the monocytes for phagocytosis, right? So these T cells, T helper cells, they are key to our immune system. They are very central to our immune system. They are essential for our immune system. If anyway we get helper T cells damaged, then our immunity does not work. Then we get immunocompromised. As I told you, in case of AIDS, HIV destroys helper T cells. So when virus destroys helper T cells, no other immune cell is formed, and whole immunity gets shut, right? So it is the role of helper T cell which activates our whole immune system. So they are very much essential for our immune system. Then we have got regulatory T cells, or we also call them as suppressor T cells. These regulatory T cells or suppressor T cells, they maintain the balance of immune reactions. They suppress the immune system from overreaction. They stop the immune system from causing, they stop immune system. They stop immune system from causing autoimmune disease. Autoimmune diseases. So regulatory T cells, they regulate the normal functioning of immune system. So if immune system gets out of control, then it starts attacking own body components, destroying the own body organs. That problem is known as autoimmune disease. So regulatory T cells, they ensure that immune system is working normally and it is not attacking its own body components. So they prevent the autoimmune diseases, right? So that was the role of killer T cells, helper T cells, and regulatory T cells or suppressor T cells, right? Similarly, we have got plasma cells and memory cells. What is their role? So what is the role of these B lymphoblasts? They produce plasma cells and memory cells. These plasma cells, they give rise to antibodies. Plasma cells form antibodies. They form antibodies at the rate of 2000 molecules per second. So when B lymphocytes are stimulated by helper T cells, they change into B lymphoblasts. Then B lymphoblasts, they form plasma cells and memory cells. These plasma cells, they give rise to antibodies. They form antibodies very quickly at the rate of 2000 molecules per second. And these antibodies, they kill the pathogen. They kill the pathogen, right? Similarly, B lymphoblasts, they also form memory cells. These memory cells, they keep the record of invaded pathogen. Invaded pathogen for varying periods of time. So they provide us immunity for varying periods of time. I told you a day before that there are some diseases for which we get a lifelong immunity. For example, measles. There is a viral disease known as measles, which we call as khasra, or which we call as holage. So measles, it is a viral disease. In Urdu, it is known as khasra. In Kashmiri, it is known as holage. So once a person develops measles, then he is not likely to develop it again in his lifetime. Why? Because once the virus enters into our body, the virus is detected, against the virus antibodies are produced and memory cells are produced. Then those memory cells which were produced against the measles virus, they remain circulating in our blood for lifelong time. So that if the virus again enters into our body, before it starts multiplication, the memory cells identify it and they get it destroyed. They get the rubella virus destroyed, right? So those diseases for which memory cells remain lifelong for lifelong period, circulating in our blood, we get immunity for lifelong periods to those diseases, right? But there are some diseases, there are some pathogens against which memory cells work only for a limited period of time. As I told you, against tetanus, these memory cells they remain in effect only for 3 months. So if we are infected by Clostridium tetani, antibodies will be formed against tetanus bacterium. Memory cells will be formed against tetanus bacterium. Once the antibodies are formed, if they kill the pathogen, if they kill the tetanus bacterium, then memory cells will remain for 3 months in our blood. And for those 3 months, tetanus bacterium cannot infect us again, right? But after 3 months, the memory cells will be destroyed from the blood. And then we may again get the tetanus infection, right? So memory cells, they keep the record of the pathogen which had invaded our body, and they remain circulating in blood for varying periods of time. Till the memory cell is circulating in our blood, we are immune from the attack of that pathogen. Once these memory cells get lost, then we are susceptible to that infection again, right? So they keep the record of invaded pathogen so that if the pathogen again enters into the body, they stimulate the other cells immediately to destroy the pathogen before it multiplicates, right? Plasma cells, they produce antibodies. And these antibodies, they kill the pathogen or they neutralize the toxins of the pathogen. So providing immunity to our body, right? So these are the types of T lymphocytes and B lymphocytes. So T lymphocytes, they differentiated into killer T cells, helper T cells, and regulatory T cells. B lymphocytes, they differentiated into plasma cells and memory cells, right? So we get two types of immunities. One is known as cellular immunity and another is known as humoral immunity. So what is cellular immunity? So we say our body has two types of immune systems working. One is known as CMIS. CMIS. Another is known as AMIS. CMIS stands for cell mediated immune system. And AMIS stands for antibody mediated immune system. So which part of adaptive or specific immunity provides us cellular immunity? We call this as cellular immunity. Cellular immunity. And we call this as humoral immunity. So which part of our adaptive or specific immunity immune system provides us cellular immunity and humoral immunity? Killer T cells, helper T cells, and regulatory T cells. They provide us cellular immunity. So because this immunity is mainly provided by the defensive cells, that is why we call it as cellular immunity. So T lymphocytes, they provide us cellular immunity. Or we can simply say T lymphocytes, they give us cellular immunity. Whereas B lymphocytes, B lymphocytes, which give rise to plasma cells that produce antibodies, and memory cells that maintain the record of the invaded pathogen. They provide us humoral immunity. We call it cellular immunity because the immunity is in the form of cells, defensive cells. We have got killer T cells, we have got helper T cells, we have got regulatory or suppressor T cells. So the immunity is present in the form of cells. That is why we call it as cellular immunity. We call it as humoral immunity. Humor means a fluid. Humor means fluid. As we say, aqueous humor. Aqueous humor, watery fluid, which is present behind the cornea of our eyes, in front of lens, behind the cornea, in the anterior chamber of the eye. We have got their aqueous humor. Humor means a fluid, right? So this immunity, it is present in fluid. Why it is present in fluid? Because when plasma cells produce antibodies, those antibodies are basically defensive proteins. And these proteins, they remain dissolved. These proteins, defensive proteins, antibodies are proteins. So these proteins, they remain dissolved in fluid. They remain dissolved in fluid. So that is why we call it as humoral immunity. So this immunity is present in plasma. This immunity is present in blood plasma. That is why we call it as humoral immunity. You may have come to know about that those persons who have recovered from this COVID-19 infection, they are asked to provide their plasma to suffering patients. So what is in their plasma? In their plasma, they have got antibodies against the COVID-19, against this SARS-CoV-2 virus, right? So they have got antibodies. Those persons who have recovered from COVID-19 infection, they possess antibodies against the virus in their plasma, right? So these antibodies, they are defensive proteins. So that is why they are asked to donate plasma, so that they can donate ready-made antibodies to the suffering patient, right? So because they remain in dissolved state, because these antibodies remain in dissolved state in plasma, so they are in fluid, they are present in fluid. That is why we call this as humoral immunity, because this immunity is present in body fluid, right? Memory cells, they also remain circulating in this blood plasma. So that is why we call it as humoral immunity. So adaptive or specific immunity, it has got its two divisions. One is known as CMIS, cell-mediated immune system, or we call it as cellular immunity. AMIS, antibody-mediated immune system, or we call it as humoral immunity. We call it cellular immunity because the immunity is mainly provided by killer T cells, helper T cells, regulatory T cells. So they are the cells which provide us immunity. That is why we call it as cellular immunity. AMIS, antibody-mediated immune system, we call it also as humoral immunity. Why we call it as humoral immunity? Because the defensive proteins known as antibodies, they are dissolved in blood plasma. So these antibodies are dissolved in blood plasma. So they are present in fluid. That is why we call them as humoral immunity. We call this system as humoral immunity or antibody-mediated immune system. So I think this is sufficient for today. We have discussed the cells of adaptive or specific immunity. So now you know what is the role of T lymphocytes, B lymphocytes. How does these T lymphocytes and B lymphocytes get stimulated? How they form killer T cells, helper T cells, regulatory T cells, plasma cells, and memory cells, and how do they provide us immunity? So that is sufficient for today. Thank you for being with me.